Thank you, Edmund. So tonight our speaker is Dr. Marcus R. Kronforst, is a professor of ecology and evolution at the University of Chicago, where his research focuses on the genetic mechanisms of evolution, particularly in butterflies. He earned his bachelor's in biology from the University of Miami and his Ph.D. in ecology and evolution from the University of Texas at Austin, followed by postdoctoral training in evolutionary genetics before joining the University of Chicago. Dr. Kronforst was a Bauer Fellow at Harvard University Center for Systems Biology. His research, funded by the NIH, NSF, has earned him numerous honors, including the NSF CAREER Award and the Pew Biomedical Scholar Award. And with that, Dr. Kronforst, it's all yours. Thank you so much for joining us tonight.
Hi, thank you very much. Thank you, Jen, and thank you everyone for inviting me. I'm really happy to be here. Yeah, I'll tell you a little bit about our research, which, as Jen said, is we study butterflies. We study a variety of different aspects of butterflies. So actually, most of our research focuses on color patterning in butterflies. So we study sort of the genetics of color patterning. We look at different species that have like a lot of closely related species with really diverse color patterns. And we study sort of during development, the genes that are turning on and turning off to make the diversity of color patterns. And over the years, we've used that as sort of a way to step out into other systems. And one of those systems we've sort of branched out into is studying monarch butterflies and migratory behavior of monarch butterflies. And so that's what I'm going to talk about tonight. I'm going to try and share my screen here. So we'll see if that works. Are you seeing my PowerPoint? Yep. Okay, great. So as I mentioned, most of our research is focused on color patterns of butterflies. So we study, a lot of our research focuses on Heliconius the genus Heliconius from South and Central America. It's a big genus, lots of color pattern diversity. We also study swallowtail butterflies, so the genus Papilio, but specifically species from Southeast Asia, where they have these amazing polymorphisms, female limited polymorphism in particular. Again, it's all focused on the genes and the genetic pathways that create their diverse color patterns. And a number of years ago, we started to wonder about whether we could use some of those same approaches, sort of digging into the genome and using natural variation to ask about this phenomenon that we observe every year here in North America, this annual migration of monarch butterflies. And so that was really sort of the motivation for this. And so my talk today is really sort of split into two sections. One is really like a genetics and a genomics component, and it's kind of detailed. And so I'll kind of gloss over some of the details there, but I will try and fill you in on what I think are the kind of really cool take-home points from that research. And then more recently, we've been doing these experiments every summer where we raise hundreds and thousands of monarch butterflies to study their actual migration behavior. And that's led to some interesting insights over the years. And so I'll tell you about some of that work. So to get started, I just want to see if this will advance. There, do you see the next slide? Okay, so before I jump into it, I'm just going to start with the acknowledgements rather than end with the acknowledgements. So my lab over the years has consisted of a lot of great postdocs and PhD students, undergraduate students, a whole bunch of people that have worked in the lab. They're really the ones that are doing the research day to day. And in particular, there's this group in the lab that we call Team Monarch that are doing this research that I'm going to tell you about tonight. So this includes a group of current and former PhD students and postdocs. And so I have to give them a shout out tonight for this great work. And of course, I also want to thank the funding agencies that have given us money over the years. This includes National Science Foundation and the National Institutes of Health. So, of course, we're very familiar with this amazing annual migration of monarch butterflies. So I won't go into great detail, but as we know, every year in the late summer, early fall, monarch butterflies in North America undergo this migration where they fly south to overwintering grounds in central Mexico. But then during the summer, the monarchs, they have a very different biology. They exist as these sort of normal butterflies. There's these summer generations of the monarch that sort of have short generation times. They don't migrate. They just live kind of like normal butterflies. So there's this incredible variation, this plasticity within the North American monarch. And this has fascinated biologists for a very long time. And in fact, it wasn't even that long ago where biologists from North America were able to discover these overwintering grounds in central Mexico. This is sort of a famous issue of National Geographic from 1976, where scientists from the United States and Canada were actually showing, oh, we have found these overwintering sites in Mexico. So what is, I think, less appreciated is that the monarch butterfly, the same species, Danaus plexippus, it exists in many other places around the world. So it doesn't just exist in North America. The same species exists in Central and South America. It exists throughout the Caribbean. It exists across the Pacific. So it's in many Pacific islands. It's in Australia, New Zealand. It also exists across the Atlantic. So there's monarch butterflies in Southern Europe and in North Africa. And in almost all these other places around the world, the monarch butterfly is sedentary year-round. It doesn't migrate. And so I certainly, before I started really studying monarch butterflies, I didn't appreciate this really sort of global distribution of the monarch butterfly and this amazing sort of natural variation in their migration behavior. So in North America, we definitely see this incredible annual migration of the monarch. And then as you look at these other populations around the world, most of them don't migrate at all. There is some migration of monarchs in Australia, although it seems to be more environmentally sort of dependent. There's fewer butterflies, they don't fly as far. So they do migrate, but it's to a lesser extent, I would say. But then many of these other locations, the monarchs are just sedentary year-round. They have these resident populations and they don't migrate at all. And so traditionally, people thought that monarchs, based on their close relatives, which are all non-migratory butterflies that are tropical, people figured, okay, well, the monarch butterfly probably originated in the New World tropics. And then as it dispersed into up from the tropics into the temperate zones of North America, it evolved migration to deal with our temperate climate in North America. And then if you look at historical records, there's sort of this very clear story from early European explorers showing the dispersal of the monarch sort of around the world. You can see from sort of the notes that European explorers made when they visited various places, like for instance, Pacific Islands. They would note when they would catalog the insects that they saw on an expedition, and then they would go back some years later and do a similar catalog, and you can sort of document when the monarch appeared in certain places. And from those records, it appears that monarchs dispersed across the Atlantic and across the Pacific in the mid-1800s. So for a very long time, we've had a good sense of, we sort of understood where the monarch butterfly likely originated and how it came to disperse across the globe. And so we set out a number of years ago to explore this question, this history of monarch dispersal, and also to study the genetics of monarch migration using genomics. So for this experiment, for this project, what we did is we collected monarch butterflies from across the globe, and then we sequenced the genomes of all these samples. So we're going to compare the genome of butterflies from across their worldwide distribution. And so here we're looking at what does this tell us about the history of the monarch's dispersal? And then can we compare the genomes of butterflies from North America and compare those genomes to butterflies from these other populations where they don't migrate and ask, where are they different in the genomes? Are there genes that pop out as being really different that tells us something about the genetics of migration? And so along with collaborators, so we didn't, when we decided to start this experiment, the way you would begin would be to go to all these locations, collect these butterflies. Thankfully, I mean, that would take a very long time, but thankfully, people have been studying monarchs for decades. And so there's a whole group of collaborators that had already collected these specimens over years. And so I reached out to collaborators who had these specimens already preserved in freezers in labs around the world. They sent us specimens. From those specimens, we extracted the DNA, did the sequencing. So we were able to sort of jumpstart the whole project with those specimens that were already collected. And then I'll just point out that we also sequenced genomes for a handful of specimens that were closely related species, other Danaus species that are close relatives of the monarch, just to sort of put all of this into some context. And so on this slide, you can see sort of the sampling on the map here where we got the specimens from. And then in this sort of like, I'm not sure if you can see my cursor on, do you see my cursor moving here? There's this sort of, it's not even a tree, but that is essentially an evolutionary tree based on the genomes that we sequence that shows how all these samples are related to one another at the bottom. And this already gives you a picture of what the relationships show us in these genomes. Basically, an interesting immediate observation from this analysis that was surprising is that the North American samples are at the heart. They're at the base of this whole radiation. And that we didn't expect. We thought that, again, that the monarch originated ancestrally from sort of Central and South America, and then they dispersed secondarily into North America. But every analysis we've done of these data show that the ancestral home of the monarch butterfly is North America. And so this evolutionary tree basically puts all the North American samples at the base, at the sort of the root of that tree. And then from North America, we get these three separate independent dispersal events out of North America. One from North America out into Central and South America, a second out of North America across the Atlantic, and then a third dispersal event out of North America across the Pacific. Another way of looking at these data, we could just analyze them in a variety of ways, looking at different population genetic statistics. One that I'm plotting here is just this measure of genetic variation in each sample. So each triangle on this little figure here represents data for one sample that we sequenced the genome for. And this is just a measurement of how much genetic variation is present in that sample. And we're just plotting it. So a point that's plotted high on this graph means that sample has a lot of genetic variation. And then a point that's plotted low on this graph means it has less genetic variation. And there is this interesting analogy to like humans. Human genetic variation is greatest in Africa, and then as you move away from Africa, there is just this clinal variation. You lose genetic variation step by step as you move away from Africa, and that's a classic population genetic signature that points to the ancestral home of humans. That tells us, among other things, that tells us that humans originated in Africa, and then as we dispersed out of Africa, we see this pattern of how our genetic variation trails off kind of linearly with geographic distance from Africa. And in the monarch butterfly, we see the exact same pattern with North America being the ancestral home of the butterfly. So the samples with the greatest amount of genetic variation are all from North America. And then as you move geographically away from North America, the amount of genetic variation in those samples trails off essentially linearly. So this is another thing that points to North America being the ancestral home of the butterfly. In terms of this question of genetics of migration, so for this analysis, then we're asking, okay, how do all these monarchs from North America, the migratory population, how do they compare to these three separate non-migratory populations? So the data are very clear that all three of these major non-migratory lineages were founded independently. So independently, monarchs dispersed south into South and Central America, and they stopped migrating. And then independently, they dispersed across the Atlantic, and they stopped migrating. And then a third time, they dispersed across the Pacific and stopped migrating. So we have these three instances of loss of migration, and then this one population that was ancestrally migratory and is still migratory today. That's our North American monarchs. So when we compare the North American population to these three populations that don't migrate, what we're asking here is, where do we see spots where all three of those non-migratory populations have changed in their genetic makeup, but they've changed in the same way? So it's a very specific thing we're looking for in the genetic signatures here. We're not just looking for differences, but we're looking for all three of those non-migratory populations to have changed, but changed in a similar way to one another. And so we go through this with various statistical tests, and it turns out that statistically, there's a substantial number of genes that pass this statistical test. So we end up with 536 genes that show a statistically significant sort of signature here, where we see that the non-migratory populations are different from the migratory population, and they all are sort of different in the same way. And this gene set is enriched for genes that are related to things like morphogenesis, neurogenesis, things that you say, oh, these could be important for migration. But I will point out that there's one spot in the genome that is just way different from everything else. And so that's what all these figures are pointing to. They're pointing to this one spot in the genome that we can key in on that just doesn't look like anything else. Basically, there's this one region, and I'll show you a bit more in a second, but this one bit that spans just 3 genes. And in this one spot in the genome that spans 3 genes, basically, all the North American butterflies are fixed for one allele. They have one version of a chromosome in that spot. And all 3 of the non-migratory populations, they haven't totally fixed an alternate allele, but a different version, an alternate allele, a different version of that section of the chromosome has gone to very high frequency in all 3 of those non-migratory populations. And it's done this independently. So this is like a very strong signature that something very interesting is happening there. That basically one version, one or more of those genes is very important. A particular version of that gene is very important in the migratory population, and an alternate version, a version of that gene that functions in a different way, is very different in the non-migratory populations. The inference here is that basically natural selection is pushing this part of the genome to function very differently between migratory and non-migratory populations. And so if we zoom in and ask, okay, what is it about these 3 genes? There's really one of those 3 genes that stands out. It's this collagen gene. Now, collagens are they're just sort of essential genes for sort of like morphogenesis, for sort of the building of an organism. But I will point out that they do have these really well-known roles in the function, sort of the development and function of muscles. And so we do think this might have something to do with sort of like flight muscle function in butterflies. And so originally, we were really excited to look at, okay, well, how is this collagen gene functioning in migratory and non-migratory butterflies? And I just assume that, well, of course, this makes sense. If we've got something related to how the flight muscles are functioning, the migratory butterflies, they're migrating. They must be very, they're strong, powerful flyers. They've got to undertake this long distance migration. They should be, expressing a huge amount of this collagen gene to fuel these powerful flight muscles. But in fact, when we look at the expression of this particular collagen gene, it's different between migratory and non-migratory populations, but it's actually different in the other way. It's expressed at higher levels in non-migratory butterflies, and it's expressed at lower levels in migratory butterflies. And we've done other experiments where we've looked at the flight, sort of the oxygen consumption of butterflies while they're, while they're just looked at their sort of their oxygen consumption, both in flight and while resting. So it's sort of measuring their metabolic rates. And this also mirrors these kind of unexpected results with the expression of this collagen gene. We see that the metabolic rate during flight is, in fact, it's higher in non-migratory populations than in migratory populations. So essentially, I always think of migratory butterflies as, being big, strong, powerful flyers. But for both this expression of this critical collagen gene and flight metabolic rates, we find that the migratory butterflies are sort of burning less oxygen and expressing this collagen gene at lower levels compared to the non-migratory butterflies, which are burning more oxygen in flight and expressing higher levels of this collagen gene. And so I think what this is all sort of, the story this is telling is that, natural selection is pushing these populations in very different directions, but it's not so much for the migratory butterflies to be big, strong, powerful flyers, but it's pushing the migratory butterflies towards flight efficiency. That long distance migration is really selecting on migratory butterflies to be very efficient flyers. And that's what's reflected in these data. So this whole sort of genetic exploration isn't really telling us about like the neurobiology and the genes that make a butterfly want to migrate or somehow navigate to Mexico, but it's definitely telling us something about sort of the physiology and the maybe some aspects of the morphology that facilitate the migration of monarch butterflies. And it's also some unexpected components because we're seeing what I think are kind of the signatures of natural selection for flight efficiency in migratory butterflies. And you can imagine monarch butterflies that aren't migrating. So all these populations that have left North America and stopped migrating, they're being pushed in a very different direction. They're no longer saddled with the constraints of long distance migration. And so you can imagine what their lives are now dominated by having to fly fast and hard to outcompete other butterflies to get to flowers for food, to get to mates. So they're sort of being pushed in a very different direction, which may be to be strong, sort of fast flyers. Whereas the migratory butterflies, because every year they have to undergo this annual migration, they're actually being pushed not in that sort of direction, but they're being pushed towards maximizing flight efficiency. Okay, so this work has ultimately, it's revealed a lot about, I think, interesting kind of take-home messages about kind of like the history of the monarch, some things we didn't expect. We've seen some things about like maybe some physiological insights about how the butterflies are adapting to these different lifestyles. Our work now is focused primarily on looking at genetic background, and here I mean source populations. So we've got these different populations, some migrate, some don't migrate, and also the environmental cues. And here I'm thinking about season, whether they're summer butterflies or fall butterflies, and how these things interact to trigger migration behavior. And so over the past few years, we've been doing a series of experiments to explore how genetics and environment influence sort of the initiation of migration. And so this is the actual behavior of the butterflies and whether they want to migrate. And I'll just point out, that time is really of the essence for this type of work because the monarch migration, this annual phenomenon, is dwindling. And so every year, scientists in Mexico do a survey of the overwintering grounds and, try to estimate the number of butterflies that are there to give us sort of a census of this migrating population. And so if you just look at this estimate of sort of the area that's covered by overwintering monarchs, and you look at that over years, you can see this clear trend of that number of butterflies decreasing. And it's actually decreasing to the point where there's concerns that, not necessarily that monarchs are going to go extinct, certainly because they're spread across the globe, or not necessarily even that monarchs in North America will go extinct. But there is, I'd say, a concern that this phenomenon of migration could eventually stop. That monarchs may continue to exist in North America, but maybe as non-migratory butterflies. And we may, at some point in the future, no longer see them migrating to Mexico. Whatever happens, it's clear that this migration, the number of butterflies that are overwintering each year, that number seems to be on a downward trajectory. And so when we're thinking about, okay, well, how do we try to help monarchs? How do we try to conserve them? There's a variety of things that have been proposed. For instance, we can plant more host plants, plant more milkweed to try and boost their numbers. We can plant more flowering plants for them to feed on as adults, try restoring their habitats or using less pesticides or herbicides. Excuse me. One thing that has come up in a variety of contexts and is this issue of raising monarchs from eggs or caterpillars to adulthood to release them in an effort to try and boost the dwindling monarch population in North America. And so a lot of people do this as a just for fun. I mean, part of it is just as a hobby, and part of it is to try and help this population that's in this pretty steep decline. And so some of the work that we've done, our work, I should point out, is really focused on trying to understand the genetic and environmental cues that are triggering the behavior. It's not particularly applied. It's quite basic research to understand the biology of the butterfly. But the results of the research have revealed sort of over the years, by accident, some information about the pros and cons of captive rearing of monarchs. And so I'm going to go through some of these experiments to just give you some of those take-home messages. So when we're thinking about captive rearing, this sort of comes in two flavors. One is rearing butterflies from commercial breeders. So it's very straightforward. You can go online and order monarchs from commercial breeders and release them at a special event. And then the alternative sort of aspect of captive breeding would just be going outside, collecting wild monarchs as eggs or caterpillars, and then raising them by hand to adulthood to release them. And so we've done some experiments on both sides of this. And it turns out that just to get right to the punchline, there are aspects with both forms of captive rearing that are problematic for monarch migration. So first, I'll walk you through some experiments that we've done with monarchs that were bred in that we reared, but were originally sourced from commercial breeders. And then I'll tell you a little bit about experiments that we did using monarchs that we brought inside that were collected from nature. So this first experiment where we were using monarchs from a commercial breeder, we weren't really focused on the commercial breeding. We were actually going to use these butterflies as a surrogate for wild-caught monarchs. And we thought this would just be experimentally easier, and we could start our experiments earlier in the year than having to collect the monarchs ourselves. So it was going to be an easy workaround. But we thought before we did this, we should first verify that the commercially bred monarchs acted like wild-caught monarchs, and it turns out that they don't. So this experiment involved ordering monarchs from a commercial breeder and then raising them side by side outdoors with wild North American monarchs in the Chicago area through the summer into the fall, and then assaying their migratory biology. So we did this common garden experiment. We've got two populations here, the commercially sourced ones, the wild-caught ones, and then we're just raising them side by side through multiple generations through the summer and fall and assaying their biology. And so when we're thinking about migration, we're obviously thinking about their behavior, whether the butterflies want to fly south, But there's other aspects to migration as well. It's not just the behavior. Migratory butterflies also have different physiology. So migratory monarchs are in reproductive diapause in comparison to non-migratory monarchs. And there's also physical, like morphological attributes associated with migration. So migratory monarchs have wings that are larger than non-migratory monarchs, and the shape of those wings is different than non-migratory monarchs. So we assayed all these aspects in our butterflies. We looked at their behavior, their physiology, and their physical traits. And so the way we test the flight behavior of the butterflies, we don't release them, but we test them in this flight simulator where we tether them in the middle of this, this cylinder. And so the butterflies think that they're flying freely, and they can rotate 360 degrees on this fixed tether. And so they tell us what direction they would fly if they were released. But meanwhile, they're contained. They can't go anywhere, but they tell us what direction they want to go. And so we fly them in this flight simulator and just watch which direction they go. And we watch them fly for 10 minutes and record their direction. And that tells us if they're a migratory butterfly or not. And so here you can see, for instance, when we tether these butterflies, the camera is sort of underneath, recording them, looking up at them. And so in the flight simulator, the butterfly can't see anything sort of on the horizon, but the top of the simulator is open, so the butterflies can see the open sky. And monarchs use the sun as a sort of a celestial cue to figure out what direction they're facing. So they use the position of the sun, their own circadian clock, and based on those pieces of information, they can figure out what direction they're facing and then decide whether or not they want to fly south. And so when we put butterflies in this apparatus, some of them are very directional. They will orient a particular direction and then just fly in that direction basically for the whole 10 minutes. And I'm just showing a butterfly on the top that's very directional. It's just flying in that direction and really unwavering in that choice. The butterfly in the bottom is non-directional. It'll fly in a direction for a little bit and then pivot and fly for a little bit and then pivot. It's just sort of, it's not really making a choice. It's just flying in random directions. And so we see all of these potential outcomes. And then the way we summarize these results is with these charts where we're looking at each black line here is the behavior. It's summarizing the behavior for one butterfly. So that is the mean direction of a butterfly. And then the length of that line is telling us the strength of the direction, how directional they were. So a long line means that the butterfly was very directional, and a short line means the butterfly wasn't very directional. So a short black line would tell us that's their mean, that's their average direction, but that they were changing direction quite a bit. And then the red line is the average for the whole group. And so what we're plotting here is North America. So the wild on the top here, the top row are the wild caught North American butterflies. So these are not butterflies that themselves were wild caught. They are the offspring and then the offspring's offspring. But they're originally sourced from wild caught North American butterflies. And then the bottom row, this is the population that's sourced from the commercial breeder. And you can see the results when we're looking at them, the generation that was raised during the late summer, and then they were interbred with one another to produce the next generation, which we flew and recorded their behavior in the fall. So that would be the migratory generation. And you can see the North American butterflies in the late summer, they're already looking a little southern in their orientation behavior as a group. And then in the fall, they are very much orienting south. All the butterflies in the North American population in the fall are basically flying south, and that average direction is quite strong. They're flying south. The commercial population does not do that, though. The commercial population in late summer, it looks like, oh yeah, maybe they are starting to orient south a little bit. But when we look at them in the fall, they're not. They're flying in random directions. So these are butterflies that have been exposed to exactly the same environmental cues. We're treating them exactly the same as the as this wild sourced population. The only difference here is that the genetics is different. The genetic source of this population is from the commercial breeder rather than being from wild caught. And they are genetically incapable of responding to these environmental cues, and it doesn't trigger the migratory behavior. If we look at their reproductive physiology, it does trigger reproductive diapause, though. We see that both populations in the summer, they have many mature oocytes, so they have a fully reproductive, they're fully reproductively capable. They have many mature eggs in the reproductive tract in the summer. And then when you look at them in the fall, both populations are in reproductive diapause. So both groups, commercial and wild caught, are responding to the environmental cues in terms of their reproductive physiology. So that's interesting. And then if we look at their wing shape, they're different. So the the shape of the wings between the commercial and wild-caught butterflies is different. And the wings, basically, of these commercial butterflies, they're not as elongated as the wings of the North American butterflies. The wing shape of the commercial bred butterflies is more reminiscent of these populations of the monarch that are from these non-migratory populations. So of course, we had these butterflies, so we also looked at them genetically because as we understood, these commercial populations were founded from North American butterflies. And so we sequenced the genomes of some of these commercial butterflies and compared them to all the other genetic data that I told you about at the beginning of the talk. And it is true, yes, that the commercial population that we were studying was founded from North American butterflies, but genetically they're just really different. The time in captivity, being reared in captivity for many generations, has caused them to genetically diverge from North America to a great extent. So at the bottom, again, is this sort of like evolutionary tree of monarchs. Again, at the base is all these North American individuals, and then you've got the 3 natural dispersal events out of North America. And that commercial population really just looks like one of those dispersal events. These commercially bred monarchs, sort of the founder effect that comes with founding a captive bred population, and then the inbreeding, I'm guessing, that goes along with keeping it in captivity for a very long time. All that has led to strong genetic divergence between this commercial population and the wild North American butterflies. And that has we don't know what the genetic changes, the critical genetic changes are, but that has had these genetic consequences that those commercially bred butterflies are no longer able to respond to the natural environmental cues that trigger the migratory behavior. Okay. So the next thing I'll talk about briefly is this issue of raising wild-caught monarchs in sort of unnatural conditions. So we've done this in a variety of ways. So we've taken wild-caught monarchs. So we're just going outside and collecting monarchs around Chicago in the summer. And then we've taken them inside, so into environmental chambers where we've tried to dial in environmental conditions that we feel like are mimicking natural conditions outdoors, to try and like dial in temperatures and day lengths that are fall-like, to try and see if we can trigger them to develop into migratory butterflies. We've also done experiments where we've taken monarchs indoors and we've raised them inside next to a window to see if that's natural enough. Will that trigger their migratory behavior? We've done experiments where we've taken monarchs and we've raised them actually in a greenhouse. So this is, we're trying to give them as much natural light as possible, but it's in a greenhouse, so it's natural light that's filtered through the glass of the greenhouse. But then we're also adjusting the temperature in the greenhouse so it essentially matches the temperature that's outside. So they're getting sort of like the cool temperature from the late summer, fall, and they're getting the natural light through the glass of the greenhouse. So it's as natural as we can imagine, except it's not quite natural. It's a little different. So we've done all these sort of perturbations to see how tweaking their developmental environment influences their migration behavior. And the take home of all this, I'll show you some data related to this first experiment, but the answer is kind of the same for all of these. And it's basically, it appears that, as far as we can tell, this migration behavior, the southern orientation of the butterflies in these flight assays, is very fragile. And when we tweak something in their development, when we remove the natural environmental cues during development, they no longer show this inclination to orient south in the fall. So I'll show you some examples here. So these are these flight orientation assays, the same things that I was showing you. So these are, again, the genetics here are the same. These are butterflies that originated from around Chicago, and then we're capturing those butterflies, and then we're raising their offspring in the summer and testing them, and then we're interbreeding those to produce a second captive generation, which is the fall generation. So the genetics here are the same, and these all stem from the wild. But what we're varying here is their rearing environment. In one case, on the bottom row here, these are all butterflies that are raised outside in like mesh cages outside. So they're exposed to all the natural environmental cues that they're going to get. And then on the top row, these are butterflies that are reared in environmental chambers where we're dialing in temperature and day length that is sort of mimicking what they're experiencing outside, except it's not. It's very artificial. And the take-home message is, again, outdoor butterflies with the right genetics, the right environmental cues, when it comes to the fall, they orient south. They want to fly south. But the incubator butterflies in the summer and in the fall, they're not flying south. We can't recreate that experience for them. And I'm not going to show the data here, but the same thing happens when we bring the butterflies inside and raise them in a cage next to a window, or even when we did it in the greenhouse. So there we were trying to get it as natural as possible, give them lots of sunlight, but sunlight filtered through the glass, and sort of change the temperature, trying to make it as natural as possible. Those butterflies also did not orient south in the fall. If we look at their reproductive physiology, there even things sort of like fell apart a little bit in this experiment. We could not get these incubator raised butterflies to go into reproductive diapause. And I will just end by saying that we might be onto something now. Just over the past couple of summers, we've continued to tweak these experiments and try new things. And we haven't published any of these results yet, but I will point out that we are now doing some different things where we are able to create migratory butterflies in the lab. And so this is very new, and it's kind of exciting. And so this experiment, we've changed a number of things. But so these are butterflies that we have raised in captivity, and we've raised them in an environmental chamber. But the difference between the previous experiments and these new experiments is that we're altering the conditions in the environmental chamber, and we're altering them on a daily basis. So we're essentially changing the temperature and the day length every day. So each day, as the butterflies are growing up in there, we're cooling the temperature just a little bit, and we're changing the day length just a little bit, so that the developing insects aren't just experiencing like a short day and a cool day, that like a standard like short day, cool day, and just setting that and saying, okay, that's the fall condition. We're giving them this gradual experience of sort of transitioning from summer to fall. So they're experiencing the gradual decrease in day length and the gradual cooling that comes with going from summer to fall. So that might be an important cue to the insect that we hadn't previously taken into account. And then the second really big change here is that instead of testing the butterflies in this flight simulator, which is very artificial, and we do it on the ground, and all the butterflies see is this little patch of open sky, we're now doing these experiments thanks to this sort of amazing work by a current PhD student, Shang Yu. She's invented this design where she attaches the butterflies to this weather balloon, and then she also attached to the weather balloon is a video camera, and then she releases the whole apparatus on this line that goes about 100 feet up in the air. And so the butterfly and the camera and a little weather station all go up in the sky. And so the butterfly is getting the full experience that comes with migration. And so they are, I think we're recreating a lot of this sort of migratory experience for the butterflies. So we've sort of changed their growth conditions, their developmental conditions, and then we're also changing sort of how we're assaying them and giving them a much more natural experience in this assay, which comes with sort of this high altitude experience. And when we do that, then the butterflies that have been raised in these unnatural conditions, not experiencing any sort of outdoor exposure, then they will orient south, even though they are effectively summer generation butterflies, but they've been raised in these sort of artificial fall-like conditions. And so that this is the first time we've ever been able to do that. And so this, for our research anyway, really opens up a lot of doors to start investigating what these cues are, and then sort of digging into kind of the molecular biology that makes a migratory butterfly. And I think that's about it. So I'm just going to conclude by saying that genetic and environmental factors are clearly interacting here to facilitate migration and really to turn the migration on. Changing temperature and day length during development, it's these sort of, my initial impression here is that it's likely this change in temperature and day length during development that are critical cues for the butterfly. And it's not just the developmental conditions. There's something about the immediate experience of the butterfly while they are flying. Here I'm thinking about things like their height, height above ground, the wind that they're experiencing, temperature, their nutritional status, all of that could be important cues that the butterfly is using when they're initiating their migration. And then all of this, of course, is dependent on the appropriate genetic background. That's very clear from all the work we've done, that the genes have to be just right for them to be able to process all of these environmental cues and then initiate migration. And that's it. That's all I've got. And I really love to take any questions that you have.
Hi. So I don't want to jump the gun, but I did have just a quick question, and I'm
Yeah.
sure you guys are still a little bit away, but this was very exciting to hear, especially about the differences in the caterpillars and raising them. Do you guys think at any point down the road you guys would be selling caterpillars?
No, I don't think so. I mean,
Just
we
curious.
we do during the summer we raise a lot of insects. We every summer we do these experiments. We raise a lot of butterflies to do them. And I just I can't imagine raising one extra one. It's such
No,
a
yeah,
it's such a
I
project.
completely get that. It was something I was thinking about.
Yeah.
But yeah, thank you.
I have a question.
Yes.
Wonderful presentation. Thank you so much. Joyce Clowley, I don't know how to put my camera on, but anyway, the question would be, say you took one of these non-migratory butterflies, say from New Zealand or
Yeah.
Australia, and brought those into your new lab
Yes.
conditions, do you think that they would orient south, or is the
Yes.
genetics of them overpowering, so that would not be possible?
Yeah, that is a great question. That, in fact, we've been trying to do that experiment for a long time. And we're always, along the way, we're finding out new things about how the environment has to be just right. There's always some hang up. So a number of years ago, we did this experiment with monarchs from Costa Rica. And so it wasn't even in the new environmental setup. The real experiment we want to do is just really, like, side by side outdoors, really giving them all the environmental conditions, the natural North American environmental conditions. What happens to these populations? Their ancestors left North America, hundreds of years ago. They have not experienced a North American, summer to fall transition in many, many generations. Do they have any latent capacity to respond to it? And so we tried this a number of years ago with butterflies from Costa Rica, and most recently we've been using monarchs from Puerto Rico. And the jury's still out, honestly. I can't answer yet. My guess is that they will not orient south, I don't think, because I think they're very much, I think that those experiments with the commercial bred monarchs, I think that really is a window into the process. Like, that is, it takes some number of generations removed from migration. So there's other things happening with the commercial bred monarchs. They're these small founding populations and the inbreeding, and some of that might happen in these naturally natural dispersal populations. But it's just, taking the butterflies, preventing them, removing the cues, the natural migration cues, and stopping them from migrating for a large number of generations. And over that time, they lose it. They're sort of the genetic basis, whatever allows them to respond to those cues, it degrades. And so I suspect that they do not have that capacity anymore.
Okay, so genetics overpowers the environmental conditions that you
Right.
might
They both have to be they both have to be there. And in the North American butterflies, they're both present. And then if one of them is not right, it doesn't happen. And in those in the commercial butterflies, and I think in these natural dispersal populations, the genetics have veered too far off that they can't respond to the environmental cues. That would be my guess.
Yeah, nature versus nurture. Oh, very interesting. Thank you.
Yes. Any other questions?
Yes, Gary Annable here. Have you looked into any of the microelectronic tags, such as those used by Icarus or CubeSat? And how would that be going?
It's a great question. We haven't done any of that. All of our work is of the type that I talked about today. We raise the butterflies, we hold on to all the butterflies. And this is, part of it is, just experimentally, that's how we approach things. And then part of it is kind of motivated by this idea that I've always been driven by the idea of leveraging the natural variation. Like, can we use these natural populations that don't migrate and compare them to the North American butterfly? And so I always knew that we would be in a spot where we'd be doing experiments with butterflies that we could never release. Like, we'd never be raising Puerto Rican monarchs and be able to release them. We can raise them, we can do experiments with them, but we always have to keep them. We'll never want to release those into the North American population, because they come from a very different place. I will say that I know people who are working on sort of these micro tags. There's a former postdoc from my lab who's worked with a team where they're developing these miniature light sensors. And so they're attaching these to monarchs every year. And then based on, I don't understand exactly the technology, but based on the light input to the tag, it's able to track the position of the tag geographically. And the only challenge there, of course, is that all of these tags have to be recaptured, have to be recaptured in order to collect the data from them. So currently, that is the big challenge with these tagging assays is collecting the data, which at the moment requires recapturing the insects. We're not at a place yet where we can put like a GPS tag on a monarch butterfly. Those are all still too big.
Oh, because some of us were in a conference at Cape May, New Jersey last month, and we watched them actually put tags on monarchs and release them. Those particular tags were being monitored. The frequency, they were solar powered, and they emitted a frequency that was picked up on a Bluetooth device.
Oh, yes, I've heard of this. Yeah.
Yeah.
So the idea is that these can be monitored remotely, but they still have to come within some distance of a Bluetooth device. Yes.
Right.
So I guess that's our best, sort of our first step towards remote monitoring. The dream, of course, is that eventually we get like miniature GPS tags where it's small enough, you can, the way you would tag a migrating bird, where you can put the tag on, release the insect, and then, sit at your computer and actually see where the insect is and track their progress towards Mexico.
Yeah, I've forgotten the researcher's name, but there's people out of Max Planck in Germany who are tracking moths and butterflies in Europe off satellites with miniature. So it could be useful. One other question that may be completely outside your realm of research, but the monarchs that are in the South Pacific and Europe, what do they use for a larval host plant? Do you know? Are there milkweeds there, Asclepias there, or?
It's a good question. I believe so. I think part of it is probably like introduced milkweeds, and then there are places, I don't know the genera, but there are relatives of Asclepias that are used as host plants in some of those places, but I'm not familiar with exactly what the species are. But I think in some of those locations there are, close relatives.
Oh, good. All right, thank you. That was an excellent presentation, and good luck with your research.
Thank you.
So let me see. So it sounds like the people that are raising monarchs themselves are probably not helping the monarch population much. And I kind of wonder, could they be doing even more harm than just the individual butterfly? Like if a commercially raised monarch is not going to be able to migrate, and it breeds with a natural North American butterfly, does the offspring know how to migrate?
Yeah, that's okay. So that's a good question. So I will say that, by far, most people that are raising monarchs are not ordering them commercially. They're going out and collecting monarchs from, around their house and then raising them. And so I think that's totally fine to do. My only advice would be to make that process as natural as possible. So, our research suggests that during the rearing of those butterflies, they need to get as natural a experience as possible. I feel like we're starting to understand what those environmental cues are, but it's still a bit up in the air. So just the more they can be exposed to the natural environment during their growth, the more likely they are to be migratory insects. Your question about sort of the genetics of the migration, we don't know. I don't have an answer for that. So whether the sort of release of commercially bred monarchs into the population are sort of detrimental, I don't know what that's doing. I will say, every once a year, there is this massive sieve, right? Where butterflies that can migrate do migrate. And so there is this constant sort of selection
Yeah.
for migration that sort of keeps selecting for that phenotype. So my guess is that it's unlikely to be a big issue because I can't see that there's that many commercially bred monarchs being released, but I don't know the answer to that. And I think just, if people are raising monarchs and they just try to do it, so my advice is just to, take, raise monarchs, do it in mesh cages, but try to put those mesh cages on a screened-in porch or outside. We want the butterflies to have sort of the temperature, the day length, all of those natural experiences. Of course, not the predator and parasitoid natural experiences, but the rest of it.
So there is one question in the chat, and it is, do other highly migratory species of butterflies also have non-migratory populations like the monarch have?
That's a great question. I don't know too much about it, actually. I believe the answer is yes. I think there are other migratory populations do have yeah, I'm sort of going out on a limb. I'm not exactly sure, but I do believe there are other migratory insects and migratory butterflies. There is some variation in whether they do or don't migrate, or direction of migration, or where they overwinter, things like that. That is an excellent question.
Are there any other questions? Or Edmund, do you have anything else? Otherwise, I don't want to hold our speaker. Thank you so much for joining us again, Dr. Kronforst.
Thank you all very much. It's a real pleasure to be here. If you have any follow-up questions or anything you think of, any comments, feel free to send me an email. I'd love to hear from you.
Thank you so much for an excellent presentation. Very informative. Thank you.
Yes, thanks. It was great.
Thank you.